My name is Randy Hecht, and I'm a GI oncologist at UCLA. And I apologize for sounding like I have gravel in my throat, but I'm doing my best. But I'm really lucky to have a wonderful panel of researchers, and both clinical as well as basic, who are really moving the needle on cell therapies in solid tumors. For a long time, we've been looking enviously at our colleagues in the hematologic space. And I think really now some of the data that was presented really is some interesting results. And I'd like to have my colleagues introduce themselves since I'm losing my voice.
My name is Saul Priceman. I am a cell therapy researcher and the director of the Cell Therapy Research Center at the University of Southern California at the Keck School of Medicine, Norris Comprehensive Cancer Center.
I'm Elliott Brea. I'm a thoracic oncologist at Dana-Farber Cancer Institute, as well as a cell therapy researcher interested in a role for cell therapy in non-small cell lung cancer.
My name is Ben Schlechter. I'm a GI medical oncologist, also at Dana-Farber in Boston. I co-direct the Colorectal Cancer Center. My main clinical interest is in immune effector cell therapies and new cellular therapies for solid tumors, in particular GI malignancies.
My name is Jennifer Specht. I'm a breast medical oncologist at the University of Washington and Fred Hutch Cancer Center. And I am the clinical director of our research program and have interest in cellular-based therapies as well for breast cancer.
Would each of you want to give a 1 or 2 minutes, just sort of summary of what you discussed today for the audience?
Sure. Most of our work has been in solid tumors in developing CAR-T cell therapies, although we've dabbled in a number of immunotherapy modalities, some of which we've taken to clinical trial for prostate cancer, for breast cancers that have metastasized to the brain, and in ovarian cancer. And then we spend a lot of time engineering those cells because, as we discussed a lot today, we know that single-targeted CAR-T cells may not be as effective for solid tumors as for hematologic malignancies. And so we're engineering them in a multitude of ways and aiming to get good clinical experience now in those settings.
Yeah, and I think building on what Saul had mentioned, we've seen that while we're getting early signals that our CAR-Ts and solid tumors are working, we've had, unfortunately, dismal results for a number of our trials, and we're trying to figure out what are the strategies we can do to optimize it. Some of the work that, I've participated in that we've been pushing forward is looking at using targeted therapy. These are, often either pills or antibody-based approaches, which we know, target oncogenes that drive the cancer. Many of these are FDA approved. Patients tolerate them very well. They have high response rates, but these patients will ultimately progress on these targeted therapies. And we're looking to see if these can synergize with CAR-Ts. And, our early preclinical data suggests that they, will work actually more potently together. It allows for the CAR-Ts to be more effective and more durable. And, then hopefully achieve what we see in some of the hematologic malignancies with our CAR-T strategies that have led to approvals, with really significant durability being seen. I think that's what we all hope for, is that we can have more of our patients getting, a one-time dose of CAR-T. Obviously, it's a logistically challenging therapy to give and often involves patients getting admitted to the hospital. But, we've talked to patient advocacy groups and, they've expressed interest in a therapy that's a one-time thing that would have a long-term ongoing benefit to them, because otherwise our hope is that the toxicities are upfront and that patients can get these long durable benefits and then not have to be coming in for repeat infusions or taking medications that could potentially have side effects that affect their daily life. And so that's what we're looking to hopefully continue to push forward.
Ben?
Yeah, and in the same vein, it was funny as I was watching or thinking about my talk, and then I saw Jennifer's talk. And we talk about the exact same challenges that we're facing in immune effector cell therapies for solid tumors, where we need good targets, we need persistence, we need expansion, we need tolerability. So the data that I presented was one approach that we're using and one of many approaches, which was this coupled CAR technology, which LIL and previously ICT has, of a CD19 CAR vector, actually 3 CD19 CAR vectors, and then an immune effector approach against mucosal target guanylyl cyclase C, where you give all the above and you create sort of a mix of different CAR cells. And some of those CAR cells are really anti-cancer cells, and some of the CAR cells are going to be just cytokine support. And some of them are this sort of paracrine microenvironment cytokine support to really overcome the limitations that we see, so that we can get in a CAR, that we can see an effective response, that we can generate a durable response, and a deep response in patients using different technology. We're all trying to find different technology to do that because it's hard. It's not that B-cell CARs are easy, but there are just different barriers in solid tumor CARs. And so, yeah, we're all talking about the same problems. We're all approaching them a little differently, but it is interesting to see that it's like we all showed up to the party wearing the same outfit type thing.
Jennifer, you want to talk?
Exactly. And I think, my role today was primarily as moderator, and I think I had the privilege of seeing some really promising data from the other speakers in our session about actual true successes and really remarkable responses in limited in glioblastoma and then in the GPC CAR realm as well. As a breast oncologist, again, we look longingly at lymphoma and other diseases where CAR-T cells and cellular-based therapies have really made a big impact. We've made baby steps, and I'm happy to say that the steps that we took sort of laid the foundation of identifying all the problems and all the challenges. But over the course of time, it's very enheartening to see how with the more widespread use of armored CARs and with really thoughtful manipulations of the tumor microenvironment that we actually are making steps forward that will ultimately really move the needle and bring this to the clinic. And finally made the point that, there's so many patients in my world that are really at need for these types of advanced therapies, even in the setting of having many, many types of different targeted therapies for breast cancer. There's still a lot of patients who are continuing to need additional therapies. So
I've been fortunate enough to be at iwCAR-T now for a number of years, and I think they've been forward thinking in having this solid tumor day. And one of the nice things is that we are seeing responses, and we used to not see responses at all in solid tumors. And the next really hurdle, and I think that y'all were talking about, is just we're not getting the persistence of the cell therapy, and we're not getting the durable responses that our patients really want. Or if we are, we're getting them in very small numbers of patients, and we have to figure out who those patients are and how we can optimize it. Does anyone else have any thoughts about sort of, everyone here is actually working on exactly that? And that, do you think that maybe in a future iwCAR-T, we'll be talking about maybe, sequencing things, or, trying to dial down toxicity, or because we're not there yet with the efficacy? Any thoughts for the future?
I would just add, I think one of the themes that came up in our session was about just intriguing thoughts about timing and when to apply the cellular-based therapies. And if we start to see more substantial proof of concept with regard to safety, as well as those efficacy signals to trying to implement cellular therapies at the right time, which is, to me, that's going to be an important step forward, because we continue to see themes of less responses in the setting of very advanced bulky disease, as opposed to maybe more minimal residual disease, such as Dr. Elliott Brea's work was suggested.
Yeah, and I mean, I think that, that point is just well made, is that the idea is that hopefully, we are testing patients with advanced metastatic disease, bulky tumors. We know that heterogeneity and antigen loss is an issue. I hope that, with earlier lines of treatment, and we're seeing this even with tarlatamab, which is a T-cell engager, that's been FDA approved in extensive stage small cell lung cancer. Now we've got trials moving that up into the first line maintenance setting, where patients been significantly debulked with chemotherapy, as well as already, anti-PD-1 therapy. But we think that might even be a better scenario. And I think that's a theme that we'll continue to see, is that, either using T-cell engagers, immune effector cell therapies, hopefully we'll even have newer modalities such as NK engagers to start show promise to really then, bring in different arms of the immune system to address the tumors. So, I'm hopeful that as time goes on, we'll have more of these therapies, and we'll have a wealth of issues of thinking of how we sequence these therapies, and whether we're, involving somebody in a immune effector based treatment as a second line option, or potentially first line.
In some ways, the more options you have, the harder it is to bring it further forward. Yeah. In some of our worst diseases, like pancreatic cancer, that it becomes easier. You have, breast cancer, as you were mentioning, Jennifer, or in lung cancer, you have many different, or in prostate cancer, you have many different, options, and it's harder because of the investment for the patient. Cell therapy, but probably also is similar to vaccines. Vaccines, having worked in the vaccine space, really have not worked in patients who are, heavily pretreated, who are last line. On the other hand, patients with MRD is probably where we're seeing our most interesting results. And so the question is, that goes back to safety as well, is that you have to have a really safe product if you're going to give it early on, and someone who might live for a while longer, or you might not even relapse anytime soon.
You actually made a lot of choices in LYL-273 about who the patients would be we enrolled. Actually, it was a pretty restrictive enrollment about the most bulky patients. Part of that was when the trial was designed, it wasn't clear how long it would take to manufacture product, and it wasn't clear because of the DLT windows, 28 days between patient, things like that, which are required in a phase 1 trial. And so the early day designs of that meant that the patients had to have a little bit more moderate disease. And I think one of the lessons of that experience, where we are seeing real responses, where we have patients more than 2 years off all therapy, which refractory colorectal cancer is, as you know, Randy, like essentially unheard of. That patient selection is important. That was done to maximize safety, and that's a reality. But I think it speaks to the lesson that we do need to move these things really early, off of the first line, something like that in colorectal cancer, or as soon as possible in solid tumors, because by the time we have patients with highly refractory disease, where in colorectal cancer response rates are in the single digits.
So that's going to be
just
for
not possible.
patient, identifying the right patients, advances in manufacturing, allo, in vivo.
I mean, even the most successful therapies we think about that are not T-cell based or immunotherapies, patient selection was such a critical component. And I think, we've learned this also with other therapies, that lower disease burden and earlier lines of therapy when they're not so refractory, especially for CAR-T cells. What we think about today being autologous, we have to take those cells out that are in the body and manufacture them, and it's the state of the host, of the patient, that's a critical factor.
I think another question that I have is that I've been mostly working with industry, but obviously most of the advances have actually come from academia, and these are very expensive. And these are very expensive trials to run. And that, how do you think that academia is going to continue to run these trials, particularly when funding is not what it has been? And or will it just be in collaboration with industry? Or
we've seen an earlier partnership with industry in our development, even at the preclinical stages, where we require that for not only design and help and forward thinking to commercialization, but also for funding. And there's also another theme that I think we could adopt, which is, do we need 12 to 18 patients on a phase 1 to demonstrate safety early, but an early and robust clinical signal that gives you, and we talked about this, about moving something to a phase 2. At what point do you see the right signal that you can move on? And I would argue we don't need 12 to 18 patients to identify that these days.
Do you think that's more for safety? But if you have efficacy in the first, several of the talks had efficacy in the first patient in a cohort, which always makes you feel good. Mm-hmm. But if you're seeing efficacy, and then that may attract the funding that you need to go big.
Yeah. I also think there are going to be a lot of changes in the manufacturing. I mean, we'll see some more in later sessions about the in vivo CAR, but that's been certainly very promising. I think the in vivo BCMA CAR that was recently presented, I believe at AACR, that's shown a lot of promise, and so that actually may change a lot of our clinical paradigm, because we're used to giving off-the-shelf therapies. It's nice to be able to dose a patient with an antibody drug conjugate a couple days later. It's very hard to with a patient that's rapidly progressing. So I think that's been the difficulty with, even in the case of small cell, we've had a DLL3 CAR-T trial. It's been very difficult to enroll to, because we have tarlatamab approved. There's no wait for manufacturing, and our patients with small cell have rapidly progressive disease. So.
But it was easier in the heme space because the prior products already worked. Mm-hmm. So now you're just trying to make it easier and bring it to the masses. Yeah. As opposed to we have to get at least somewhere near their efficacy and durability.
There's target pylon effect, and we certainly see this with her too, right? Mm-hmm. Where there's a good target and everyone's jumping on it, and there'll start to be problems with the target, but everyone's already committed. And rather than seeing that as a sunk cost, moving on to new targets, I feel like both in academia and our industry partners stay committed to the same targets. And so it was also nice today to see some novel targets and some novel techniques, whether that's armoring with IL-12 or whatever it was, to not just jump on the really important targets, but actually be pretty creative. And I think we do need some creativity to take the next step in these therapies, because these are hard diseases and hard challenges to overcome.
Thank you very much for your attention. I think we're really fortunate to have some of the leaders in the field in some of the most difficult to treat solid tumors. I think today's session really showed that we really are seeing responses. And I think that after years of really depressing results with cell therapy, I think that these results are interesting enough that I think it offers hope to patients. And I think it also may open up additional funding that we need to really bring the field forward. Thank you very much.